This is my first video on enzyme inhibition. This will cover the basics of enzyme inhibition. Our learning objectives are to interpret Michaelis-Menten and Lineweaver-Burk plots, distinguish between competitive and non-competitive inhibitors, tell how irreversible inhibitors work, describe feedback inhibition and allosteric regulation, describe the functions of kinases and phosphatases, list various ways that enzymes are regulated, tell what isoproteins or isoenzymes are, and give a diagnostic use. Inhibitors. They limit reactions such as blood coagulation to the site of injury. If you cut your finger, you wouldn't want all the blood in your arm to clot, only you want it to clot right at the cut. Neutrophil elastase is involved in phagocytosis and killing of bacteria inside neutrophils. If it gets outside, it's a big problem. Drugs. Statins are used to control cholesterol synthesis in the liver, and we'll talk about those later. A lot of drugs are enzyme inhibitors. The HIV reverse transcriptase inhibitors have done away with AIDS and made HIV infections a chronic condition. Penicillin kills gram-positive bacteria. So here's competitive inhibition. In competitive inhibition, the inhibitor binds in the active site of the enzyme. And you can form an ES complex, which breaks down to enzyme plus product, or you can form an EI complex, but you cannot form an EIS complex. So here you get to form the substrate complex, which would normally go to product, or the inhibitor, which cannot go to product and stays there. So what happens if you do a Michaelis-Menten plot? In the Michaelis-Menten plot, you get the blue line if it's uninhibited, and here's Vmax. You get the red line with the competitive inhibitor, and that changes the apparent Km because it changes the equilibrium here with the substrate, because this is going back and forth. So eventually this red line will approach the same Vmax because you can add enough substrate to push that reaction all the way up to Vmax and crowd out the enzyme, because if you make S very large, then you can't form this down here and you'll only have the normal, this reaction going. Now if you look at the Lineweaver-Burk plot, you will see that you get a different Km, a different minus 1 over Km intercept on the x-axis, but you get the same intercept on the y-axis, so 1 over Vmax doesn't change. So Km changes, but Vmax doesn't. So keep that in mind. Non-competitive inhibition. Non-competitive inhibitors bind to a separate location on the enzyme, therefore changing the enzyme and its reaction with the substrate. So it slows the enzyme reaction. So you can still have the ES complex product, but you can form an EI or an ESI here. And both of these are dead. So essentially you're reducing the amount of enzyme that can ever be made into product, because this one can't go to product either, the ESI complex. So if you look at the Michaelis-Menten plots, you'll see that again we get the blue plot with no inhibitor, but we get a green plot with a non-competitive inhibitor, and we get a lower Vmax. So Vmax has changed. No matter how much substrate we add, we can't go back up there because some of our enzymes locked up down here. So we essentially reduce the concentration of enzyme that we had in the reaction. Now Km is unchanged because this equilibrium has not been affected. And so, but Vmax is, and so you can see that Vmax has changed, minus 1 over Vmax for the no inhibitor, minus 1 over Vmax for a competitive inhibitor. Irreversible or suicide inhibitors, inhibitors that irreversibly react at the active site. Also, suicide inhibitors are substrate-like molecules. Michaelis-Menten and Lineweaver-Burk plots are not used to analyze these. They lower the concentration of enzyme. They essentially kill the enzyme and lower Vmax without changing Km because I is not competing with S. Suicide inhibitors are special cases of irreversible inhibitors in which the inhibitor is acted on by the enzyme to form a covalent EI complex. A great example, classical example, is penicillin. Enacts the enzyme that cross-links D-alanine peptides and glycine in the cell wall of the bacteria. of gram-positive bacteria, such as Staphylococcus aureus. And so here is the enzyme. Here is penicillin, and it reacts with an active site serine to form this acyl complex here. And so this is an irreversible reaction, and this complex is there, it's enzymatically inactive, so it's dead. Allosteric enzymes also do not follow Michaelis-Menten kinetics. They are oligomeric enzymes with multiple subunits, as you can see here. Here's an example with 2 subunits here. And there are positive effectors that are activators, and you can see an activator plot here. Or you can have a negative effector, such as inhibitors, and you see what the effect of an inhibitor here is. This is the no activator, no inhibitor reaction in black. They are homotropic effectors, or substrates. Heterotropic effectors are non-substrate molecules. So think of sigmoidal curves, and remember hemoglobin and O2 binding. They function very similar to that. A lot of them are used in metabolic regulation steps and metabolism, and you'll see a lot of this from Dr. Robinson and others as we go through the semester. Feedback inhibition is quite often involved in allosteric, because we may have an enzyme that is enzyme 1 that's converting A into B, and then B can go to either C or F, and finally other products. And then B may feed back on A. Once you've made enough B, you want to slow this down and not have too much of it. Also, E may feed back on enzyme 2 to reduce the amount of E being formed, but not affect the amount of G that's being formed in the other side of the reaction. So it's very interesting, and feedback inhibition is a major control mechanism for enzymology and metabolic pathways. Activation and inhibition via post-translational modifications. Zymogens, such as trypsinogen, made by the pancreas. It's made as a zymogen, inactive protein molecule, and it's only activated once it gets into the intestine, in the duodenum, where it's activated to the active enzyme trypsin. If it was activated in the pancreas, it would digest your pancreas. Proenzymes, such as prothrombin. Prothrombin, you don't want thrombin making blood clots all the time, so you make it as a proenzyme, and so you convert it into thrombin only when you need it to clot the blood. Phosphorylation. Kinases add phosphates to proteins, sometimes to activate them, sometimes to inactivate them. Dephosphorylation, the enzyme is phosphatases. Phosphatases take phosphate groups off, and that can do an activation or inactivation process. So here's an enzyme that with ATP, protein kinase, makes the enzyme, puts a phosphate on there, and then we have a phosphoprotein phosphatase that takes that phosphate off. You can get activation or inactivation, and since it has to have a hydroxyl group, the enzyme can have a serine, a threonine, or a tyrosine. All 3 have hydroxyl groups that can be phosphorylated and dephosphorylated. And that's the end of that part. Thank you.